Emily, and I am the head of communities here on TuDiabetes, working with the Diabetes Hands Foundation. And it's time for our Thursday live interview. I'm so glad to see we have a bunch of people in the chat room anxiously awaiting information on the liver's role in diabetes. So our guest today is Bob Geho, who is the co-founder and CEO of an organization called Diasome Pharmaceuticals. Bob, welcome, and thank you so much for joining us today to present to us the role of the liver in diabetes. I think this is not talked about nearly enough. So the camera is on you, and thank you so much for being here. Can you please start by telling us a little bit about yourself?
Sure. Well, first of all, Emily, thanks very much to you and your colleagues at the Foundation for inviting me. I'm delighted to have a chance to talk about diabetes in general, but specifically this afternoon, as you say, the role of the liver in diabetes. I completely agree, and my colleagues at Diasome agree that it is it's a part of diabetes understanding that we think is not talked about nearly enough. So I'm delighted to be here. My background is in life sciences, drug development. I've spent about 20 years working in various aspects of the pharmaceutical world, and a lot of that time has been focused on working in pharmaceutical development and diabetes in general, and then more specifically in trying to figure out how to get drugs like insulin to work better once they get inside a person, either via injection or also via an oral administration. And so today what I'd like to do is spend some time talking about the liver and its role in glucose metabolism, but then also talk about why does insulin work the way it works right now, and are there strategies that can be employed to make insulin work better, in addition to all of the important things going on with insulin analog development and artificial pump and artificial pancreas and closed-loop pump systems. So that's a little bit of my background.
Fantastic. Then let's actually go ahead and launch right into your presentation of all of those things. We already do have a question in the chat room that I think you'll probably get to later, and that is to talk a little bit about liver-related complications of diabetes, which tend not to be discussed that often.
Right. Right. Well, we can certainly get into that in the course of the conversation. So I will go ahead and get started here. So one of my current favorite philosophers from the last century or 2 centuries ago is a physician named William James. He kind of tongue-in-cheek asked, is life worth living? And even back in the 1800s, people seem to intuitively know that, in fact, it all depends on the liver. I'm not sure that it all depends on the liver, but in many respects, there was a lot of wisdom in that answer. A healthy liver is critically important, and part of what we're focused on at Diasome is trying to get livers as functional as they possibly can. So I have 3 important questions that I want to use to kind of frame the discussion today. The first is, how exactly is glucose managed in a healthy individual? Because we think that understanding how normal glucose metabolism actually occurs is critically important, not only for researchers in the realm of diabetes, but also for patients. I find more and more as I interact with people who have diabetes, and for full disclosure, I'm a type 1 myself, so I'm very comfortable talking about the various aspects of diabetes care, but I find that as I interact with people who have both type 1 and type 2 diabetes, there can be an information gap in terms of just understanding how glucose is actually managed in people who don't have diabetes. So that's the first question I want to address in these slides. The second is, why is it that injected insulin acts differently than pancreatic insulin? To put that in a little bit of context, in a healthy adult individual, their pancreas is producing, generally speaking, anywhere from 18 or 20 units of insulin up to 40 units of insulin in a 24-hour period. And it's probably not an exaggeration to say that the average type 1 diabetic, if there is such a thing, is injecting a lot more insulin than even the high side of what's normal. So 50, 60, 70, maybe even 100 units of insulin a day. And so the question is, why is it that insulin is acting differently after injection than the insulin that comes out of a healthy pancreas? And then finally, what exactly happens when insulin is targeted to the liver in people with diabetes? So with those three questions in mind, let's look at some very important data that was generated all the way back in 1978. You'll notice that in the course of these slides, I really like using, I'm not sure it's considered old data, but older data. So this is from a leading light in diabetes research, Dr. Ralph DeFranzo from Texas. And this is a paper that he published in the Proceedings of the National Academy of Science in 1978. And without getting too much into the weeds on this, the point of this slide is to show that when glucose is taken orally, the liver dominates. So they looked at healthy individuals, and they gave them, in the first three bars, glucose that was administered peripherally, meaning they gave an intravenous injection of glucose. And what they were looking at was how much of that glucose is actually taken up or stored by the liver. It's referred to in the legend as splanchnic glucose uptake, which is the scientific phrase for liver uptake. But you'll notice on the far right-hand side that when glucose is given both intravenously and orally, the liver almost goes into hyperdrive and stores glucose in a very profound way. The reason why this is important, and we'll talk about this in a couple of slides in more detail, but one of the reasons why this is important is that this shows that glucose that is given peripherally doesn't behave in the body the way that glucose that's taken orally behaves. And that's really important because that has a huge impact on the way diabetes drugs are analyzed, both in academia and in industry. But the point I want to make here is that most of us, unless we're in a hospital situation and are getting intravenous infusions of glucose, most of us on a daily basis, whether we're healthy individuals or have diabetes, get our glucose orally. And when we do, and if we're healthy, our livers are storing an awful lot of that glucose. So here's what happens in normal metabolism. And if you think about the physiology of the GI system and the endocrine system, the pancreas, as we all know, is producing 24 hours a day, 7 days a week, 365 days a year in healthy people, a lot of things, including insulin and glucagon and other very powerful hormones. The pancreas is connected to the liver via blood flow that's called the portal vein. And in response to rising glucose levels, on average every 4 minutes, the pancreas is pumping either small amounts of insulin if we're not eating, or very large amounts of insulin if we are eating a lot of glucose, directly into the liver. And it's the liver's job to use anywhere from 50 to 80% of that insulin. And in response to that insulin signal, the liver stores at any given time during a meal or a snack 30 to 65% of the glucose that we eat if we're healthy individuals. And so the liver is acting like a reservoir, or some people have used the analogy that it's like a sweep account that you would have in a banking system where you have a lot of cash coming in and then you sweep cash out into other accounts on an as-needed basis. But in this case, the liver is acting as a sweep account for glucose, because the reality is when we eat typical carbohydrate loads, we're taking in a lot more glucose than the peripheral blood needs. And so the liver's job is to act like the kind of court of first resort to stop that glucose from just invading the peripheral blood and overwhelming the peripheral vascular system. And then the liver, in a very sophisticated, very elegant system, is able to release both insulin and glucose and other things into the peripheral blood. And by doing so, healthy individuals have, generally speaking, tightly controlled peripheral glucose, and importantly for the discussion today in the TuDiabetes community, very rare occurrences of hypoglycemia. Okay, so this is, in some respects, a very simple view of liver metabolism and liver glucose uptake, but it does set the stage for the rest of the presentation, and I just can't emphasize enough how in a healthy individual the liver is, number one, keeping most of the insulin that the pancreas produces and releasing very small amounts, relatively speaking, of insulin into the peripheral blood, and it's keeping a big chunk of the glucose that's taken in when we drink or eat meals or snacks. So in terms of normal glucose control, there are three primary cell types in the body that have insulin receptors. The first is the liver hepatocyte. These are called hepatocytes, where they're hepato meaning liver. These are the cells that get hepatitis, for instance, which is why hepatitis is called that. And as I showed in the prior slide, they use up to 80% of all the insulin that the body produces from the pancreas. The other two cell types are muscle and fat, or it's otherwise known as adipose. But importantly, only hepatocytes can both store and release glucose into peripheral blood on demand. So fat cells and muscle cells, in response to insulin in peripheral blood, will take glucose out of the blood and lower peripheral blood glucose, but they can't release glucose back into the peripheral blood to service the heart and the brain in response to hypoglycemia. So one of the things that we're going to talk about is that when we inject insulin now, we're primarily hitting only muscle and fat cells. It's not to say that none of the insulin that's injected gets to the liver, but the dominant part is going to muscle and fat. And so that creates what we call a biodistribution problem with injected insulin therapy. So it's our view, and we think this is strongly supported by key research that's published in the literature, that the liver, in fact, is the #1 site of glucose control in a healthy individual. And that's primarily because of the things that I've just outlined and emphasized here, that the liver hepatocytes are designed to both store glucose as glycogen and then in an on-demand manner release glucose back into peripheral blood. And one of the reasons it's kind of interesting to think about the development of artificial pancreas systems and closed-loop systems. It's very engineering focused, obviously. And I think that in some ways that's a response to the recognition that the system of the information loop that goes from the pancreas to the portal blood, to the liver, to the peripheral blood is extremely sophisticated. And while we may be frustrated with the long duration of the development cycle for these new artificial pancreas systems, I think the challenges there are in direct relation to the just absolute elegance and sophistication. It's almost mind-boggling how the liver and pancreas are able to communicate with each other and store and release glucose in response to that communication. So I want to talk a little bit about a brief history of insulin development. Obviously, insulin was discovered in 1922 by Banting and Best in Toronto. You could actually buy animal-based or animal-sourced insulin up through 2005 from Eli Lilly. And then regular recombinant human insulin was introduced by Lilly and then by Novo in the 1980s, and that's still available today. And then, of course, in the mid-90s, Humalog, and then quickly thereafter, Novolog, as the first rapid-acting insulins came online, and then that led to the development of the long-acting basal insulins. And so the progression of insulin development has really gone from the animal sources to the first biotechnology version to the more sophisticated analogs. But the primary development of these insulin therapies has been once we jumped from the animal-sourced insulins to the biotechnology products, they've been focused on onset of action, getting insulin so that it would come out of the subcutaneous tissue after injection either faster or slower than regular insulin, and then manipulating via chemistry the duration of action. So the two dominant aspects of insulin development have been speed of onset of action and then duration. And of course, this is big business. It's estimated by Cowen and Company that the global insulin market will be a $26 billion a year business by 2018. But I want to emphasize that back in 1972, so 42 years ago, a researcher at Columbia University made what we consider to be a significant but largely ignored discovery. And that discovery is summarized in this slide. This gentleman's name is Robert Canfield. He was a senior researcher and the head of the Irving Center for Clinical Research at Columbia University in the early 1970s as a chemistry experiment and not so much as a diabetes experiment. He made the first radioactive insulin molecules that still had all of their biological activity. So in other words, he was able to make insulin that still worked like insulin after injection into an animal that was radioactive, so he could find it in these animals after injection. And what he found, much to his surprise, and you have to remember this is 50 years after the discovery of insulin, and he was really the first person to show and to publish that subcutaneously injected insulin did not show up in the liver. So he could find the insulin molecules in fat cells, in muscle cells, and in the heart, but he could not find any insulin in liver hepatocytes. And so in discussions with some of our senior researchers going back to the mid-1970s, late 1970s, and early 80s, there was a lot of discussion at the time about whether or not you could find a way to redirect insulin molecules after sub-Q injection to liver hepatocytes. So the question inevitably is, well, why is it that insulin doesn't reach the liver in a more normal fashion? And we believe that it's simply because there are so many insulin receptors in fat and muscle cells that when we inject insulin subcutaneously, that insulin is just picked up by all the insulin receptors in the periphery, and it makes it very difficult for the injected insulin to reach the liver. Now, you can just keep increasing insulin doses to try to drive more insulin to the liver, but we all know what happens when we go out of bounds in terms of escalating our insulin dose. The risk of that is often hypoglycemia. So one of the dose-limiting realities is that we can't give enough safely with current insulins to get insulin back to the liver. Now this may, depending on the experience level or the amount of reading or interaction with physicians and researchers that the audience today has, this may be news, but it's interesting because I went to the artificial pancreas Wikipedia page a couple of days ago, and I had never looked at it, and I thought in preparation for this discussion I would just pull it up and see what it said. And I've highlighted and read some of the interesting comments. So they first of all say that there are notable differences with insulin replacement, meaning how we inject insulin today, compared to the function of pancreatic insulin delivery. In the second bullet point it says pancreatic insulin is released into the portal vein, which is what we've just talked about, and it does that so that the liver, which is, as it says, is the major organ for storing glycogen, can store glucose. But if you go down a couple of bullet points, it says insulin, which is not delivered intravenously, cannot achieve normal momentary concentrations in the portal vein, which connects the pancreas to the liver. And so replacement insulin is dosed as a best compromise between aggressive use for lowering the blood sugar when eating, but also being conservative to avoid post-meal low blood sugar. So this information is out there. But as I said at the outset, I still find that it's not widely discussed, as you mentioned, Emily, at the beginning. So, all right. So here's our diagnosis of insulin. Because the liver in a living system, meaning a human being or an animal, is very inaccessible. And by that, what we mean is that there are really only two ways to figure out how drugs like insulin work on a liver in a living person or in an animal study. We either have to do very invasive abdominal surgery, which has a lot of complexity and some danger associated with it, or we have to do very esoteric kinds of catheterizations, where we put a catheter in through a peripheral jugular vein. It actually has to go down through the aorta and into the portal blood and up into the liver. And it's very hard to get ethics committee approvals at research institutions here in the United States and also around the world to do those kinds of studies. So in a sense, most, if not all, of the drugs that have been and are being developed for treating diabetes, whether they're insulin or other things, are done without factoring in how they affect the storage of glucose in the liver. Now, that's not to say that none of that work is done without the liver being taken into consideration, but it is a significant problem simply because from an anatomy point of view, the liver is very inaccessible. And so 30 years ago, 35 years ago, most early stage research on glucose lowering drugs began to focus on the use of what are called peripheral glucose clamp models. And peripheral glucose clamp models are viewed by the diabetes research world, both in academia and in industry, as the de facto gold standard for how we assess whether or not diabetes drugs are working in people. And the way they work at a very high level is, instead of giving oral glucose to animals or people, we inject, as I said in the very first Ralph DeFranzo slide, we're infusing glucose into a peripheral vein and then administering drugs and assessing how those drugs lower the glucose that we put into the peripheral blood. The question is, so what? Well, the so what is that 30 or 35 years of medical research has focused on the role of muscle cells in glucose control, not on the role of the liver. Because in peripheral glucose clamp models, it's much easier to assess the effect of diabetes drugs on peripheral muscle uptake of glucose than on the role of the liver. And our view is that that's a very important and somewhat negative outcome. So to give you an idea of this, this is, I call this is like my Kramer versus Kramer slide. I call this DeFranzo versus DeFranzo. So Dr. DeFranzo published a paper that was under the Lilly lecture in 1987, so 8 or 9 years after the first study that I showed. And this is a study where he was looking at how glucose is controlled in both, or where it's taken up in both control patients who are healthy individuals, and of course NIDD is type 2 individuals. And what he showed is that when glucose is infused peripherally in healthy individuals and in type 2 diabetics, the muscle, not the liver, which would be splanchnic at the top, the muscle is the dominant part of glucose uptake. So this has had, in our view, a direct impact on the way the liver has been viewed over the last 30 or 35 years. I would encourage people watching this to ask their endocrinologists, just out of curiosity, where they think the dominant part of glucose uptake occurs in individuals. And I would say that it wouldn't be any surprise to me, at least, that the common response would be reflected in this chart, that the dominant action of glucose uptake in individuals is in the muscle. But again, as we showed in the very first chart, in direct distinction to this, where the glucose is given peripherally, if you give glucose orally, it switches from muscle glucose uptake to liver glucose uptake. All right, so insulin therapy today. Intensive insulin therapy, as we all know, is hampered by the risk of hypoglycemia. Closed-loop artificial pancreas systems are challenged by both hypoglycemia and by insulin's onset of action. And that's understandable. When we think about closed-loop systems, to the extent they're trying to mimic normal pancreatic insulin release, if you go back to that first flow chart, the insulin is coming out of the pancreas on average every 4 minutes. Insulin has a very short half-life, and so there's this direct pulsatile effect of insulin that's primarily acting not in the periphery but in the liver itself. And so we're trying to mimic that with better hardware in the form of CGM and better pumps, and we're also trying to mimic that with ultra-rapid acting insulin analogs. And all of that is trying to address this fundamental challenge of mimicking the pancreas and the liver. So, as I said, therefore, global development, both in academic medicine and in industry, is focused really on speed and duration on the insulin side, and then on the algorithms and the hardware for closed-loop technologies. But we think that hepatic insulin, meaning liver insulin biodistribution, is still generally not recognized as either a necessary, or if it is recognized as being necessary or desirable, that it's not achievable. Because, as I said, there are so many insulin receptors in peripheral fat and muscle cells that getting insulin past that barrier from an uptake point of view to the liver is a big, big challenge. So here's what happens with current insulin therapies in our view. When we inject insulin peripherally, not all, but the majority of that insulin is picked up in peripheral blood. Very little of it, meaningful amounts, essentially don't reach the portal vein, and that means that the liver is not able to store the 60 to 65% on the high side of the glucose that we eat as diabetics. That leads to poor peripheral glucose control, frequent hypoglycemia, and chronic hyperglycemia. You know, it's the liver's job in a healthy situation to be the first defense against hypoglycemia, again because liver hepatocytes are designed to release glucose into the blood to counteract hypoglycemia, and they do that in response to glucagon signals from the pancreas and epinephrine and other things. So in our view, the preferred form of insulin therapy would be to have both portally available insulin and peripherally available insulin. And that if we could do that, the liver would begin to function in a more normal fashion and patients would have overall better glucose control. So what I want to do now is move to a discussion of how we would actually achieve that and what happens, to answer my third question at the beginning. You know, what happens in individuals, whether they're animals or human beings, if we do provide hepatic or liver-specific insulin? So the way we do this is we've made a material called HDV, which stands for hepatocyte-directed vesicle. It's a 20 nanometer carrier that binds both liver-targeting molecules. So we have a library of molecules that are very safe and can be embedded into this carrier that are specific for receptors on hepatocyte cells in the liver. And then that carrier will bind to insulin molecules. So we can inject, for instance, liquid HDV into vials of insulin. We have a rapid-acting insulin here and a long-acting insulin, but it can be worked with. It can work through any class of insulin. And what it does is when patients inject themselves either with a pump or with a syringe, and you can also do this orally, it redirects some of that administered insulin so that it goes not only to muscle and fat, but now also to liver hepatocytes. So here's what happens when we do this. This is a study that was done in diabetic rats. So we made rats that were, you can make them chemically so that they're completely insulin deficient. But before we did that, we were measuring C14 labeled glucose. So you can make glucose molecules radioactive, and then you can track where they go in the liver. And you can see that the black bar here on the left-hand side is how glucose is taken up in the liver of these animals before the animals are made diabetic. So this is kind of normal liver glucose storage. After the animals are made type 1 diabetic, you can see that even though they're getting glucose, that glucose is not being stored at nearly the same rate as it was before the animal was made diabetic. So this is the problem with diabetes in large measure, the liver, which is being measured here, is not able to store glucose normally. We then gave the same animals glucose and an injection subcutaneously of Humulin. So this is regular human insulin from Eli Lilly. And you can see that it did very little to increase the amount of liver uptake of glucose. But if you add the HDV targeting material and you give the same total dose of insulin to these rats, you can see that all of a sudden we get a dramatic increase in liver glucose storage. So the mechanism of action for liver-targeted insulin primarily is to move individuals, in this case diabetic animals, back to the normal range of hepatic glucose storage. This next study is a study that was done in dogs that started out as healthy, intact animals, and we gave them a standard oral glucose tolerance test. So this is the same kind of test method that human beings get when they go to the doctor. If the doctor thinks that they might be either a type 1 or a type 2 patient, you're given something to drink that has a lot of glucose in it, and then the physician will check the blood glucose level every 15 or 30 minutes. So the green curve that's kind of in the middle here is what normal dogs' blood glucose looks like after a standard glucose challenge. So they become slightly hyperglycemic up through 110 or 120 minutes, and then they begin to level out at their starting point. These animals were then made type 1 diabetics, so they had no insulin production of their own. They were given the same glucose challenge and a standard subcutaneous dose, just like people would get, of human insulin. And you can see that they became fairly dramatically hyperglycemic up through the first 120 minutes, and then they became they began to head towards hypoglycemia out at 150 to 180 minutes. So the blue line is really the standard curve of people with diabetes. And what we try to do with insulin therapy and with other oral anti-diabetics in the case of type 2 patients is try to get that blue curve so that it squeezes down to be more like the green curve. But we wanted to know what would happen in these same animals if we gave the same glucose challenge, the same total dose of insulin in each animal, but we added the HDV material to target some of that injected insulin to the liver's hepatocytes. And the red curve is what happens. And you can see that we were storing more glucose in the liver because we gave the same total dose of glucose, but less of it was showing up in peripheral blood. And so our assumption is that the glucose has to be in the liver because there's really no other place for it to logically to be. And that's evidenced by the fact that we're then releasing glucose out towards the end to counteract hypoglycemia that could otherwise be caused by the tail period of the injected insulin. So really, all we want to do with hepatic targeted insulin is take people who should be at the green curve but are experiencing the blue curve and get them back down to the red or the green lines. So it's a fairly, at that level, it's a fairly simple premise. We're just trying to reactivate the liver's normal glucose storage mechanism so that it can pull glucose, keep glucose from going into the blood immediately after a meal, so in the 30 to 90 minute range, and then release it to counteract hypoglycemia as it appears to do in the 180 minute range. So then the question is, well, what happens when we do this in people? But before I do that, this was a long-term study. This is going to speak to, well, what happens with hypoglycemia? So this was a long-term study that was done in dogs. This was done in Edmonton by the same people who were involved in the original islet cell transplant work. And we had dogs who had been pancreatectomized, so they were completely insulin deficient. They had no ability to produce insulin. And they were given 1 week of either regular insulin therapy or 1 week of HDV insulin therapy as their only pre-meal insulin. And then we shifted them to 2 weeks on regular insulin and then 2 weeks on HDV insulin. And we're looking at their average blood glucose levels throughout the time of day, so starting at 9:30 in the morning and going through 6 o'clock late afternoon, early evening. And what you can clearly see is that even though the animals were given the same total dose of insulin throughout the study before each meal, the addition of the hepatic targeting component dramatically reduces their overall plasma glucose levels. Now, these dogs were running on the high side because they were extremely diabetic given the pancreatectomy. But the point of this was to show that there was a fairly dramatic decrease in overall glucose levels in their peripheral blood. And then when we looked at their reduction in mean daily plasma glucose on the left-hand side, that's almost 100 milligrams per deciliter drop when we compared the regular insulin treatment weeks to the HDV insulin treatment weeks. And importantly, we were able to reduce hypoglycemic events very dramatically. And that would make sense physiologically, because if the liver is able to normally store glucose in a type 1 situation, it should be able to normally release glucose to counteract hypoglycemia. So now we're going to look at what happens in a group of type 1 human subjects. This study was done on our behalf at Vanderbilt University, and we had 9 well-characterized, very typical type 1 patients. They had no C-peptide, so they weren't able to make any insulin on their own. We gave them an oral glucose challenge, just as we did in that dog study, and we gave them a standard subcutaneous dose of human insulin. And the blue curve is looking at the change in arterialized blood after the glucose and insulin administration. And you can see a fairly hyperglycemic curve there. We then gave the same patients the same glucose challenge and the same total dose of injected insulin, but we added the HDV material to the insulin. So even though they got the same total dose on a per unit basis of insulin, we were able to reduce the peripheral blood glucose by almost 38%, simply by adding a targeting material that redirects a very small amount of insulin to the liver hepatocytes. So our goal with this therapy would be to have patients use their regular insulin therapy, combination of short-acting pre-meal insulins and basal insulins, but have HDV added to those insulins to generate this kind of overall blood glucose reduction meal after meal, day after day, for the balance of a person's life. The other thing, my last data slide, is that we measured peripheral insulin levels in the patients in the study that I just showed in the previous slide, and there was no difference in the peripheral insulin levels, which we would expect because most of the insulin in HDV insulin is free, non-targeted insulin. So we're able to achieve the effect in this chart with no change in peripheral insulin action. It appears to all be hepatic-specific or liver-specific effects. So to recap, in healthy people, 100% of the body's insulin production goes first to the liver, and 50 to 80% of that insulin stays in the liver. In healthy people, the liver stores anywhere from 30 to 65% of a typical glucose load. And yet, in most drug development, even today, peripheral glucose studies focus primarily on glucose uptake in muscle for all the reasons I've outlined, number one of which is the liver is just very hard to get to. And in the real world, a significant percentage of oral glucose should be taken up by the liver, because that's been demonstrated by Dr. DeFranzo and many other people that oral glucose really does go to the liver first. So here's what I characterize as insulin users' Bill of Rights. Here's what I think insulin users should be aware of and should be pressing for from the diabetes development community with respect to insulin specifically. My view is that all injected insulin therapies should be hepatic-specific or liver-specific, because all normal pancreatic insulin is first of all hepatic-specific. It's just basic biology when we look at it from that point of view. I also think that injected insulin should protect against both hyper and hypoglycemia because pancreatic insulin protects healthy individuals against hyper and hypoglycemia. And I think that, and this sounds overly simplistic at some level, but I think it's worth saying, I think chronic insulin therapy should be more than life-saving. It's not enough to say to a person with diabetes, well, if you want to lower your blood glucose, just take more insulin. I mean, we who take insulin know how wrong that is from a mindset point of view. And we who take insulin and take other anti-diabetic agents know the complexities, the day-to-day complexities of glucose regulation that our friends and neighbors and relatives who have healthy endocrine systems don't have to put up with. So that's the end of my formal presentation. I really appreciate the opportunity to present this to you, and I'd love to try to answer any questions that the audience might have.
Bob, we have a host of fantastic questions because that was an amazing presentation. I think people have learned a lot and, as a result, have some great questions. So I'm going to kind of go down my list here of questions that I've been compiling as you've been speaking. And the first one is from Lloyd, who is asking if you would please explain how metformin works with the liver or affects the liver.
Yeah, that's a terrific question. So metformin is unusual in the class of oral anti-diabetics because it actually inhibits, we think, I mean, it's still a little bit of an open question from what I understand from medical researchers in this, but the best conclusion I've heard is that metformin's primary action is to inhibit liver glucose production. So the liver does two things. On the one hand, in a healthy individual, the liver will store glucose, but the liver can also release glycogen as glucose, and it can produce glucose. So metformin, from what I understand, has as its primary mode of action the inhibition of what they call hepatic or liver glucose production. And that's in distinction to the majority of the other type 2 diabetes drugs, which are either insulin secretagogues, meaning they are pressing the pancreas to produce more insulin, or insulin sensitizers, which are obviously designed to try to make peripheral cells more sensitive to the insulin that the body's producing in the context of a type 2 subject. But it's a great question.
And a very good answer. So our next one, this is a question from David, who's asking, who's saying liver complications don't seem to get mentioned and discussed to the same degree as others such as eyes and kidneys. Can you talk about those complications a bit?
Yeah, so I'm not an expert in liver complications, but what I can tell you is that one of the questions that we get in the context of the information that I've just presented is, are questions related to, well, will the livers respond, number one, in the case of a person who's had type 1 diabetes, for instance, for a long time, 5 years or 10 years or 20 or 30 years. And what we found in human clinical testing is that the liver has a remarkable ability to in a sense, have the lights turned back on and begin almost immediately to store glucose in a normal fashion or near normal fashion. Now, there are situations with fatty liver and cirrhosis and other hepatic impairments where, for instance, from a clinical point of view, we're going to have to do clinical studies to look at that and see how hepatic-specific insulin would work in those situations. But I think our general assumption as it relates to our view of hepatic-targeted insulin is that the liver will respond in a near normal fashion or normal fashion, even in patients who have had diabetes for a long time. At the same time, I do think, and I went back and I watched the presentation that Christy gave quite some time ago from Columbia University on the role of the liver in diabetes on the TuDiabetes website. And you know, she said that she had tried to find information about type 1 in the liver, and it was very difficult to find. And I actually responded to Emily about that and said, yeah, it doesn't surprise me that it's hard to find. I think, to kind of round out my response to my answer to the question, because the liver is so hard to get to anatomically, it is really hard to find information about this. And so, to David's question, I don't see that changing anytime soon.
So you mentioned fatty liver just now in that answer, and we actually have a question about fatty liver from Kalupar, who says, I have a concern about fatty liver. Is it expected for those like myself who have type 2, and especially for folks who are overweight?
Yeah, so, you know, I'm not a physician, so I would hate to say whether it's expected in your particular situation, but I think that it certainly is a concern with type 2 diabetics. And a lot of that has to do with the fact that, in our view, there are probably multiple things going on in liver metabolism and liver glucose uptake that have not so much to do with insulin per se, but with other things. And so I think, for instance, people like Alan Charrington of Vanderbilt, who we view as one of the liver's rock stars, if you will, has done a lot of work and published on what he calls the portal signal. And it may be that there are signals other than insulin that have to be present in order for the liver to store glucose normally, and it may be that type 2 diabetes is in some respect directly related to a deficiency of whatever that portal signal is. And so the problem is we get into these things that physicians and researchers call hormonal cascades, or cascades of the various enzymes in the liver. So once one thing goes wrong in an organ like the liver that's trying to metabolize or store large amounts of glucose and deal with fat and all of that, it's very easy for things to get out of whack very fast, and the domino effect can be very, very complicated. So again, I would hesitate to say a whole lot more about that in terms of an individual type 2 and their situation with fat accumulation in the liver. But I think that at the very least, it should be thought about and discussed with a physician, because for sure the liver can get out of whack, and when it does, all kinds of things can go wrong.
All right, our next question is from Dishers, who says, I have found out that I have an enlarged liver and I have to see a specialist about it. Could that be related to cholesterol medication? Slightly different topic, but you may know.
I would say, Emily, I'm going to take a pass on that. I probably shouldn't comment on that. I'm not sure.
Completely fair. So then the next one, we have a question from Kate, who's saying, prednisone was a major culprit in speeding up my type 2 diagnosis due to rheumatoid arthritis. How does the diabetes plus the prednisone adversely affect the liver? Do you know about this one? This is a toughie.
That is a toughie. It's also a good question, but I hate to pass on two questions in a row. It kind of goes against my own rule.
No, it's fine.
I'm going to take a pass on that as well.
It's fine. These are super good questions that are slightly off topic for this particular talk. So we have a question from David about HDV, the product from Diasome. He's asking, how far away is HDV from being generally available? We had a lot of folks in the chat room commenting on how exciting your research is, and when can we have it?
Yeah, well, thanks very much. So HDV has been what's called enabled for phase 3 testing by the US FDA. And so we're planning on running our first pivotal phase 3 study in type 1 patients beginning in early 2015. And we're just really excited about the potential impact of HDV, first of all, in the type 1 population. We think that it could be a significant breakthrough in making already available, already commercialized insulins like Humalog and Novolog and Apidra to get started, and Humulin and Novolin as well, just much more user-friendly. So it's a great question. We're hoping that if all goes well, we could be on the market assuming our NDA is successfully viewed by the FDA in the 2017, early 2018 timeframe, if everything goes exactly as I have it mapped out in my brain.
And we hope it all goes exactly as you have it mapped out in your brain.
Right.
So now we have a question from Dr. Jody who's asking, is the only benefit of HDV insulin better blood glucose control? What about more ability for the liver to respond to a low or less weight gain from injected insulin due to more glucose going to the liver as opposed to adipose cells? Or any other improvements in liver function due to having more glucose or fuel?
All of those are great questions, and we think that HDV or some other type of hepatic-specific insulin would have a positive effect on all of those. So I'll try to go through them systematically. In terms of the liver's ability to address hypoglycemia, we think that the mechanism of HDV should be relatively straightforward. The way a healthy individual addresses low blood sugar in the peripheral blood is that the liver releases glucose that's been stored as glycogen, or it produces new glucose through gluconeogenesis. And all of that is happening in the liver itself, and the glucose that has been stored at the time of a meal is stored in direct response to insulin in the liver. And so we think that in long-term human testing of HDV insulin in type 1 patients, our hope certainly is that the liver will be able to store glucose on a much more normal fashion and therefore be able to release glucose to counteract hypoglycemia on a much more normal fashion. So that's the first thing. I think that it should have a big impact on hypoglycemia. The second thing is that it could be possible that the addition of HDV to commercial insulins will enable patients to take less insulin overall. And that would make sense if the liver is now storing anywhere from 1/3 to 2/3 of the glucose that's taken in. That means there's less glucose in the peripheral blood that has to be dealt with by peripheral insulin. So we would think that there would be a high likelihood that the overall amount of insulin that's required will go down. If that happens, we would then think that the typical weight gain that goes along with aggressive insulin therapy should be mitigated. I can't say by how much, but it would make sense that if we're reducing the exposure of peripheral fat and muscle cells to insulin because of a better hepatic response, that's just less glucose that's going to be taken up by fat, which stores glucose as fat. So, if we think about it, hopefully not in too simplistic of a way, we would think that it would have at least a weight neutral or best case scenario, a positive effect on weight gain or weight loss. So great questions. Did I miss something there, Emily? Was there one more that is a part of that?
I'm rereading the question to make sure you got all of it. No, I think that you hit on all the pieces.
Okay.
Here's a really interesting question from Terry in the chat room, who's asking if you have any comments about Afrezza and the concept of inhaled insulin in relation to insulin's action in the liver.
It's a great question. So obviously Afrezza would be the first, aside from the Exubera product from several years ago, would be the first non-injected insulin. It has a very rapid onset of action, presumably because of the large surface area in the lung that's very absorptive. Our view is that the onset of action is very positive, but that insulin still has to be hepatic specific. And whether we absorb insulin in through the lung or we inject it peripherally, it's still going to have a challenge at getting to the liver. So we haven't done studies on inhaled insulin with hepatic targeting. It's something that we would love to do. And I would just say in general, I'm always encouraged by novel developments that are going on in insulin. So I'm really excited about the advisory committee's response to Afrezza, because I think the more of that we see, the better off we're all going to be.
Rick has a production question about your product, which is about HDV, which is how can it be mass produced at high volume? Does it need its own whole factory? Can it be sort of produced along with the insulin that it would be injected with? How will that piece of the puzzle work?
Yeah, also a great question. So HDV has been designed to be manufactured in a very straightforward way. It can be manufactured with pretty typical pharmaceutical manufacturing equipment at very large scales. The good thing is it's not exorbitantly expensive. I joke with people that because of the work I do, I'm very much in favor of high pharmaceutical prices, but I have to talk out of both sides of my mouth on that. So it's very cost effective. Let's just put it that way. And the interesting thing about it is that it can be done in two different ways. And we've interacted with the FDA on both of these strategies. The first strategy would be a very traditional situation where we're collaborating with an insulin producing company and they add the HDV at their insulin manufacturing site before it goes into vials or pen cartridges. The other way we could do it, which has a certain amount of appeal to many of us at Diasome, is that the HDV could be sold in a standalone fashion and could be added by either the pharmacist or the patient directly to either their pump cartridges or vials of insulin, which would be a very novel way of thinking about insulin therapy. I mean, we're on the one hand, we're all used to, depending on how long we've had to inject insulin, used to mixing. If you talk to your parents, for instance, if they were a type 1 diabetic, mixing long-acting and short-acting insulins was just part of the routine. We now have pre-mixed insulins, but the idea of adding something to insulin that's not insulin, that could be what we call a functional excipient as an admix product, is very feasible and very doable. The good thing about that is it would enable more people to have access to the HDV system.
We are almost out of time, but I have a couple more questions that I'd like to ask you, if it's okay.
Sure.
We might end up going a couple minutes over time. Thank you. So
Sure.
One is VTOR is asking, does this therapy, HDV, mean that I could look forward to having a return to near normal energy levels once I have a storage of glucose in my liver?
Well, what I can say is that certainly is the hope. I mean, we're very focused as a team here on using or viewing insulin really as hormone replacement therapy. Insulin has a relatively straightforward action on cells, starting with liver hepatocytes and then moving to fat, muscle, brain, and other cells in the body. And it just has to be there at the right time in order to enable these cells to do what they normally do. And so, at the risk of being overly simplistic, we really think that, especially in insulin-using type 1s and insulin-using type 2s, that the healthier we can make glucose metabolism in the hepatic region, the better overall health people are going to have. And certainly that would be evidenced by greater ability to do the things routinely that healthy individuals do without the fear, for instance, of hypoglycemia and without the just ongoing problems of elevated blood glucose that tend to slow us down. So that's a long-winded answer to say, yes, I certainly hope so. I hope it makes people feel a lot better.
That would be fantastic. I just like the concept of returning to near normal energy levels. Since I've had diabetes since I was a tiny little kid, I'm not sure how different that would feel for me, but I like the idea.
Right.
And our final question is from Linda, who is asking if the liver is the culprit in our morning high blood sugars.
Well, I guess it depends on where you were when you went to bed and a variety of things. One of the perverse aspects of diabetes, especially in type 1s, is that, as I said, the liver, and this is well known, this is not unique to Bob Geho saying this, but the liver, in addition to not storing glucose in a diabetic situation, is also really good at producing glucose. And so the way a healthy individual's liver is stopped or told not to produce glucose is from an insulin signal. And so that is one of the real problems. I mean, people call it the Donna factor or other things, but one of the real problems is in a healthy individual who's fasting or sleeping, the pancreas still knows when glucose levels are beginning to rise because of hepatic glucose production. And so the pancreas will release insulin into the portal blood, into the liver to stop that or slow it down to bring people back into normal glucose ranges. In the case of a type 1 diabetic, if there's not insulin in the liver is going to be tricked into thinking that the person is becoming hypoglycemic, and they'll just start it'll start making glucose from fatty acids. So yeah, the answer is yeah, a poorly insulinized liver is a culprit in that, in the Donna factor, elevated morning glucose levels.
Darn.
Yeah.
You work on that wonderful product.
Well, we will keep at it. We'll keep at it. But thank you very much for having me, Emily. It's been a pleasure to speak with you, and I appreciate the questions. And I'm going to go back and check into prednisone and a couple of other things, because now I'm curious myself.
Ah, wonderful. Thank you so much for joining us today, Bob. This has been really informative, and we have folks in the chat room saying that they're going to be studying this video and watching it multiple times to get a full understanding of what you have shared with us, and that is fantastic. So thank you so much for your time and your energy and your great work.
Thanks. Well, thanks everybody for watching, and I'm always available for questions, so you can go to our website and shoot me a note, and I'll try to get back to you as quickly as I can. So thanks very much.
Cool. Take care.